good. Okay, well, thank you very, very much for inviting me. It is a pleasure to be here, and I actually gained 12 or so hours, so I'm more rested than I would otherwise be if I had lost it, I think. So I actually started out my existence as an experimental scientist and spent quite a bit of time as an experimental scientist. So when I do my mathematical stuff, I'm realizing that it would be helpful if I actually had my talk up there. So when I do mathematical things, first of all, I am not a mathematician, but mainly I'm an experimental scientist, so what I do is I try to do experiments, but I do experiments using math. You can manipulate systems much more readily, easily, and more quickly, of course, if you do it mathematically using modeling. But that's not all I do. What I really do is I try to take disparate ideas and bring them together into what I feel are more coherent wholes. So this is where I do at least some of my thinking. This is Sedona, Arizona, which has nothing to do with Ohio State University, but I live here as often as I can, about 3 months out of every year. So this summer, I came upon a quote in a translation of a paper from 1962, 1963, almost as old as I am, where the author was talking about the importance or growing resistance against antibiotics by bacteria, and how even 10 years ago they had this sense that bacteria were growing resistant to antibiotics. And this author proposed that, in fact, phages may be a solution to the problem of antibiotic resistance. And the interesting thing is that we're talking 50 plus years ago when these ideas were being introduced into really the area of phage therapy. So my talk is Applied Ecology of Phage-Mediated Biocontrol, and this is what applied ecology is. So this is from Wikipedia. So applied ecology is a subfield within ecology which considers the application of the science of ecology to real-world, usually management problems. So really, when you're working with phages in environments, or even phages in infected human beings, you're talking about an ecology, and if you're using phages to control bacterial populations, you're talking about an applied ecology. I gave a talk in Yanji, China, about, it's going on a month and a half ago now, and before the talk it was suggested to me that I might want to introduce who I am, since nobody in the audience necessarily will know. And so I thought, well, I'll just put my books up there. So I put my books up there, and I actually have 3 more that are currently in progress. One of them is sort of done now, or 3 more edited volumes. When they're done, no more books, except if somebody wants to edit a volume and wants to have me as kind of like a star who does nothing. Or if I'm writing a monograph, that's fine, but no more edited volumes. So for my talk in Zurich this summer, I decided that I would go through my papers and try to figure out what I've published and what I've published on, and I grouped them together into parts. And so this is my earliest work. This up here is my first paper in 1989, where I'm looking at lysis, and I'm looking at the evolution of latent periods, and also lots of models that involve phage population growth. And the talk I gave in Zurich is actually right there, that YouTube video, if anybody wants to write that down. I'll pause a little bit. That didn't exist as of this morning, so that's why it's not in the brochure. So from going through phages and trying to understand the field broadly, editing all of these books and this background in ecology, I was asked about 5 years ago to write a chapter that's called Communication Among Phages, Bacteria, and Soil Environments. And I really did not want to write this chapter. Even then I was getting tired of writing chapters. But I thought to myself that with the idea of communication really being interactions, I could think of all of the ecological interactions that we have between phages and bacteria, their host bacteria, or phages and environments, or indeed between bacteria and their environments. And I came up with this big giant list here, which I'll get back to. So slightly later than this, starting in 2003, I published my first paper on phage therapy. And in fact, the APSNet paper is right there. It's not really a paper, it's more of a web page, but whatever. I got involved in phage therapy thinking. And since that previous slide was, in fact, put together, I've actually published all of those different papers. So I've been really getting heavy duty into phage therapy. So it seemed obvious to take this thing and convert it into phage therapy ideas, which really forms the genesis, the basis of this talk I'm giving now. So the first thing I had to do is update this. So I took out all the communications and replaced it with interactions, and otherwise did various changes. And then I simply grayed out all the areas that I thought were not necessarily relevant to phage therapy. And so you end up with something that's, there we go, with that. And I'm not going to talk about this in any length right now. If we have time, I'll get to this at the end of the talk. But this really is the basis of my thinking. We can understand to some degree the ecology of phages. We use phages as these biocontrol agents. It's an applied ecology. How can the ecology of phages inform the applied ecology of biocontrol, and how are the two different from each other? So this is just the phage therapy one. And so if I click here, so all the ones that aren't phage therapy pharmacology things just disappeared. So I've gotten involved in phage therapy pharmacology. My wife actually went to pharmacy school for 2 years, and so she has this pharmacy background, and this kind of motivated me to start thinking about phages in pharmacological terms. And so suddenly I've become the pharmacologist in the household, not her. But I've published a number of papers where I've thought about this. And this is a paper that just, or from a paper that just came out this spring. And it's really just looking at the idea of phages as drugs, in this case from a very ecological as well as physiological perspective. But when we have phages, I mean, they're antibacterials. Subset of antibacterials are disinfectants. Disinfectants you can use on living flesh or antiseptics. You have these antibiotics that are selectively toxic, and then among selectively toxic antibacterial agents are the bacteriophages. So bacteriophages as drugs. And so that's me. So taking all this idea of applied ecology, let's apply it to phage therapy pharmacology. So the big picture is that the phage ecological objective is to find target bacteria, to amplify their numbers, and that typically occurs at the expense of bacterial fitness. And then they have the new phages that are produced, find new bacteria, and so on and so forth. And that's a little different from our objective in phage-mediated biocontrol, where the object is, in fact, to cause the death of bacteria, for phages to find bacteria and eradicate or eliminate them. So these two approaches are not identical. So they're not necessarily identical, and they're not necessarily even fully compatible. So we can't be thinking solely in terms of phage ecology. Oh, phages can go into environments, phages can wipe out bacteria, therefore we can apply any phage to any environment and wipe out bacteria. So in thinking of these ideas, I decided that there are 4 key ecological factors that we should be thinking about when we think about either phage ecology or applied ecology. And so this is the first one, and that is bacterial density. So bacterial density is a very important determinant of how fast phages, their virions, can find bacteria. The more bacteria around, up to a point, the faster phages will be able to find those bacteria, specifically the bacteria the phages are able to absorb to. So this in turn is an important determinant of phage generation time, how long it takes to go from an absorption until the progeny phage are absorbed. And it's also important for what I call effective burst size. The longer a phage has to wait until it finds a bacterium, the more likely that phage is going to become inactivated. And together, these are important determinants of phage population growth, which is important in some cases in terms of phage-mediated biocontrol, but not all. In terms of that biocontrol, the applied ecology, we can consider bacterial densities that are low, high, or very high. When they're low, we employ something called passive treatment, which we'll get to. When the bacterial densities are high, we employ something called active treatment, or at least we can. And when when bacterial densities are particularly high, we might have to employ something that I describe as active penetration. And we'll get to what those are. But bacterial density is an important component of ecology and applied ecology of phages. And so is phage density. Now, in the wild, the phage density is important, at least in part, because the more phages you have around, the more phages that are going to absorb to individual bacteria at the same time. And if you have multiple phages absorbing to individual bacteria at the same time, things change in terms of phage-bacterial interactions. So you can have recombination between co-infected phages. In terms of lysogeny, lysogeny may be induced more readily when you have more than one phage infecting a bacterium than not. So that's ecologically. In terms of applied ecology, as well as ecologically, in order to have lots of bacteria being killed off by phages, you need a lot of phages, you need high phage densities. And thinking about applied phage ecology, how do you get high phage densities? You either have to add lots of phages, or you could add your phages, and the phages can actually amplify their numbers in situ while they're attacking bacteria. And then this increase in phage numbers where your target bacteria are is reduced by dilution effects, just simply adding phages into an environment, you're diluting the phages, and also any phage inactivation that can occur. That's number 2. Number 3 is the performance of the phages. Now, whether or not the phages need to perform at a high level, or what level they need to perform at, is dependent upon the conditions under which you are treating. And we'll get to those. But these are all phage-resistant mechanisms that are displayed by bacteria. And these phage-resistant mechanisms you can think of as means by which bacteria degrade the performance of phages. And this degradation can be in terms of how fast the phages can attach to the bacteria. It can be in terms of how many progeny, baby phages, that the phages can make once infecting. And it also can result in increases in generation time. So bacterial resistant mechanisms have the effect of degrading phage performance, which can be important in certain circumstances and less so in others. And this I stole from my Zurich talk. We also have phage penetration. The phages have to be able to reach the bacteria, and bacteria can be in places that phages have difficulty reaching. One of these places, which is what I'm illustrating, is within bacterial microcolonies. If you have a big clump of bacteria, it might be easier for the phages to infect on the outside of this clump than it is on the inside of the clump. And it might be difficult for phages to reach the bacteria inside the clump. Phages have difficulty reaching bacteria that are found inside of other cells, or intracellular bacteria. So penetration is just a general term that I use to describe the ability of phages to reach the bacteria physically that they need to attack, absorb to, in order to eliminate. So this says phage penetration in target bacteria and or into bacterial clumps. Those are the 4 basic ecological ideas: the number or density of bacteria, the density of the phages, the performance of the phages against these bacteria, and then also the ability of the phages to just get within the vicinity of the bacteria that you're treating. This came from a chapter that actually came out this summer in what I call the Pole book, or the Gursky book. It's a book on, in fact, phage therapy, current research and applications. So what I'm doing here is I'm combining concepts of a general scheme of phage-mediated biocontrol of bacteria. And so the first one up here, absorption, actually, in terms of treatment of plants, we don't have to worry about, because that's movement of phages into blood or drugs into blood. So I've crossed that out. Distribution is actually the movement of drugs from the blood into body tissues, but distribution can have other connotations than that. So I'm partially crossing that out. And then I'm just taking those 4 basic ecological ideas and mapping them onto here. So penetration is 4. You've got to get to the bacteria. Once you've gotten to the bacteria, you have to be able to perform at some level, and these are basically a description of that performance. The more phages you have around, the faster you'll have absorption. And then we can divide up the type of therapy that we're employing in terms of the bacterial density. Active treatment requires higher bacterial densities than passive treatment. With passive treatment, at a minimum, the phages are going to have to adsorb to bacteria. With what I call active penetration, at a minimum, the phages are going to have to be lysing bacteria that they're infecting. And then active treatment actually requires the phages to actively infect cells, effecting phage release. So we can walk through some of these ideas in terms of the phage attacking of bacteria, perhaps eliminating them, and how ecology differs from applied ecology. So this is the most basic idea. So we've got a certain number or density of bacteria. The phages are able to infect the bacteria and increase their numbers, and maybe they'll increase their numbers sufficiently so that they can substantially reduce bacterial densities, what I'm calling inundative phage densities. So this is phage ecology, more or less. But in order to have these inundative phage densities, you have to have what can be described as winner bacterial densities, as in the concept of kill the winner. If you've got sufficiently prevalent bacteria, they can support phage population growth at high enough densities that the phages will be able to eradicate the bacteria. If you don't have enough bacteria around, that's not going to happen. And that's shown here. So this is a lower number of phages. This is the number of phages, sorry, a lower number of bacteria. This is the number of phages or bacteria, the density of phages of bacteria. So when you don't have enough bacteria around, you're not going to be able to support phage population growth to high density. So we have this X here. And instead, if you're going to eliminate the bacteria, you're going to have to supply the inundative phage densities from without. You're going to have to explicitly add these and those densities to the population of bacteria that you're treating. So this is non-winner phage densities, and now we're getting more along the lines of applied ecology. These are exogenously supplied phages. So remember, applied ecology is a subfield within ecology which considers the application of science ecology to real world, usually management, I probably should have underlined that, questions. So it's applied ecology that's going on there. And these ideas of having to supply these phages perhaps in relatively high densities without relying on their replication may also be relevant to phage treatment of biofilms, but we're not quite there yet to fully understand how that occurs or needs to occur. Now, if we are not adding these phages exogenously, then we're going to end up with treatment failure. And treatment failure can be a consequence of there not being enough bacteria around to support sufficient phage population growth or insufficient phage performance. So the phages may not be able to replicate sufficiently, or in fact, they may not be able to kill bacteria sufficiently. This really is a host range issue. And so we could describe failures that come about because there are not enough bacteria around or insufficient phage performance as applied ecology failures. So I've said a lot about passive treatment and active treatment. Passive treatment is where we overwhelm the bacteria by adding lots and lots of phages, so many that we don't rely on the phages. We don't have to have the phages actually replicate in situ. Whereas with active treatment, we're relying on active phage replication. We don't add a whole lot of phages. The phages actually replicate when they infect bacteria, and then the resulting phages go on to infect new bacteria and ideally wipe out the bacterial population. So passive treatment, all this requires, other than that the phages actually penetrate to the target bacteria, is for the phages to adsorb and then kill the bacteria. However, for active treatment, you have to have lots of metabolizing bacteria around, otherwise you'll never boost the phage density to high enough densities to get rid of the bacteria. And also the phages have to adsorb, they have to infect, they have to lyse the hosts that they're infecting and amplify their own numbers at the same time. Performance requirements are much higher. And this is kind of the same thing in tabular form. So passive treatment, you have to have viruses that are able to survive well enough, both before you add them to an environment and once they're there. The phages, certainly in both cases, passive and active, have to be able to absorb to the bacteria. In fact, they have to be able to penetrate as well, which isn't shown here. But in terms of having a decently short latent period, with passive treatment, you can have a latent period of infinity. It doesn't matter. All you need to do is kill the bacteria. But with active treatment, the latent period matters. Burst sizes. With passive treatment, doesn't matter what the burst size is. You don't need to make more phages. But with active treatment, you do. In order to have lots of phages produced, you have to have sufficient densities of bacteria. But with passive treatment, it doesn't matter. With passive treatment, all that matters is the density of phages you add, not the density of target bacteria. You do have to have, however, bactericidal either infections or absorptions in order for passive treatment to be effective. But you don't have to have infections. However, you do have to add enough phages so that you inundate, eradicate the bacteria, that you have enough to wipe them out. Same thing as adding high enough concentrations of drugs under any circumstance that you can have a positive effect. In the case of active treatment, however, because these phages are actually increasing their numbers in situ as they infect bacteria, you can have much more localized inundated densities of phages. So you could have the wiping out of phages, of bacteria very locally as a consequence of very localized increase in phage numbers. So this is something I put together to try to sum up the whole idea of ecology versus applied ecology, active treatment versus passive treatment. These would be the truly natural situation where you have endogenous phages. If there are enough bacteria around, which is what this line up here is showing, then you can have substantial phage population growth, and that might reduce the bacterial density somewhat. At the other extreme, you have passive treatment, where you in fact can have not many bacteria around at all. So this is the density of bacteria in the environment that you're treating. And in fact, not much phage population growth. This would be low population growth. This would be high amounts. And still wipe out the bacteria. In between, we have active treatment, where presumably we're adding more phages than would naturally be within an environment, but we're relying on phage population growth to increase the density of phages to the point where it wipes out the bacteria. This is only possible if you have enough bacteria to begin with. And then there's something in between that comes from this paper, Payne and Jansen, 2003. I'm quite influenced by the papers by these guys. I've read them over and over and over and over again, and ultimately have gotten a lot out of them. But they have this idea of passive-active treatment. And I think that passive-active treatment, although it's kind of obscure, really is how we try to do phage therapy generally. We add enough phages so that we can more or less wipe out bacteria that we can easily reach. And then we also have some phage population growth that adds more phages, perhaps very locally, that succeeds in killing off more of the phages. And as I initially conceived this, I had a bunch of numbers up here, and I didn't put it in here for the sake of not confusing everyone. But we also have this idea that you're probably in phage therapy situations or biocontrol situations adding many, many rounds of phages, especially in phage therapy in the clinic, where you might treat a person 10, 15 times or more. But as you reduce the bacterial population, as you're more and more successful, in fact, adding lower amounts that would normally allow active treatment may no longer be effective. And you might have to actually increase the number of phages that you're supplying in order to ultimately wipe out the bacterial population enough that the immune system can get it under control. If you're not trying to reduce the bacterial population to zero, you may not have to come in ultimately with as many phages or add phages as often. But you're going to have to add enough phages so that you can at least supply inundative densities that will reduce the bacterial population as much as you need to reduce that population. So the real question is, if some phages are good, are more phages better? And the flip side of that question is, how much do we need to rely on the individual performance of these phages? If we're relying on active treatment, we're going to rely on the performance of the phages much more so than, in fact, if we're relying on passive treatment. So we move on to something else that I've been doing. This is only starting in 2005 or so. The first paper actually is 2007. And this is thinking about phages in terms of spatial structure and the formation of phage plaques and the attacking of biofilms using phages. And so what's spatial structure? It's really inhibitions on the ability to move. And so here just showing inhibition on the movement of a bacterium. Bacterium formation in cellular arrangements where the bacteria are not separating from each other physically, spatially. The formation of bacterial microcolonies, many cells that aren't separated from one another. The formation of phage plaques within these lawns of bacteria that in fact consists of many microcolonies and so on and so forth. It's really some inhibition on movement, particularly of bacteria, but also of phages. And we can think of the use of phages to attack bacteria within spatially and structured contexts. And particularly biofilms, and there are four basic ways that this occurs. This is actually a paper that might be published by now. I sent in the final proofs on Monday. I don't know how fast they've gotten this thing out, but it should be out very soon, I hope. It certainly has a PubMed entry at this point. At any rate, you can physically remove the biofilm. In fact, there are a number of animals and eukaryotes that physically remove biofilms. You can chemically remove a biofilm by destroying the extracellular polysaccharide, which is what EPS depolymerases do. You can use phages to kill the bacteria from within, and also you can supply endolysins, which are the lysis proteins of phages from without, to destroy the bacteria from without. So different ways of attacking biofilms. That's really the applied ecology. The ecology is much more complicated than this. I like to think that what happens with phages and biofilms is that a phage is diffusing around and happens upon a biofilm, particularly, if it's lucky, a clump of cells in the biofilm that are of the same type. It starts infecting those cells and making new phages. The phages may be able to diffuse short or slightly longer distances within the biofilm to find new clumps of cells that it can attack. And the phage progeny can diffuse out of the biofilms and diffuse away so that they can actually find new biofilms to attack. So a phage is specialized in attacking biofilm bacteria. Its job is to find a biofilm, start infecting, and produce as many phage progeny as it can so that there's some possibility that one of those phages may find another biofilm to attack someday. So ecologically, the issue is how readily can phages find new clumps to exploit. And this is also from this Chan and Abaddon paper. Ben Chan is I called him my pseudo postdoc. He's doing a real postdoc now in Paul Turner's lab at Yale University. But this is the idea. It occurred to me that at steady state, if you're going to have a phage population whose size is not changing, then if a phage is able to find a biofilm and start making phage progeny within that biofilm, at steady state, the number of exploitations, localized exploitations of biofilms should not be increasing. And that means that no matter how many phages are produced per focus of biofilm infection, on average, at steady state, only one phage is going to find a new biofilm to infect and start this new focus of infection. So this is our phage. This phage is the same phage as that phage there. It's going along, it's finding a biofilm, and it's starting to make phage progeny. Some of those progeny are diffusing out of the biofilm to find new biofilms. Now, if this is a pond, say, the biofilms are going to be on the surfaces of the pond, but there's going to be this huge volume within which these phages are diffusing around looking for new biofilms. So even if you're making lots of phages per focus of infection, at steady state, on average, only one phage is going to survive to make establish a new focus of infection. And no matter where you are keeping track of the phage doings, you're going to, at steady state, everything's going to be staying the same. So no matter how many phages are made per focus, you're only going to have one that survives to establish a new focus. Now, if the phage population is increasing, that will be different. More than one phage will survive. But eventually the phage population may become so big that the bacterial population becomes lower and you go back to steady state. And alternatively, if you have fewer than one, the phage population will be in decline. So that's my explanation ecologically for what goes on in terms of phage-biofilm interactions. Now, in terms of the applied ecology, what's really important is this phage, at least I believe, this phage propagation within microcolonies. So in applied ecology, the issue is mostly the phage potential to penetrate into clumps or biofilms and to do so in sufficient titers or generating sufficient titers there that they successfully kill the bacteria. So how do you do this? Either you're going to have to supply high phage titers, or the phages are going to have to perform well so that they produce lots of phage titers in situ to get rid of the bacteria there. Or perhaps we're going to supply the phages multiple times so that we ultimately are getting rid of the bacteria. Or maybe we're going to augment things with more than just phages. Since I think in phage terms, I'm thinking of indolysins or EPSD polymerases, but it could also be chemical antibacterials. If you're trying to get rid of the bacteria, you've got to get whatever you're attacking the bacteria with to those bacteria in sufficient densities and for sufficient lengths of time that the bacteria are eliminated. So returning back to phage therapy or phage-mediated biocontrol of bacteria. I've been putting together this chart. I have to update it because I now know that there's more of these than I have listed here. This is the number of publications per year. These are the experimental phage therapy or phage-mediated biocontrol papers. And you can see that there's been this steady uptick. We're going up, not quite 2 per year. Well, actually, now that I know about more of them here, maybe we are at the point of 2 or so increase per year. We had some kind of lull going on here. I'm not sure what was going on. And so with that kind of approach to doing things, I figured as long as I'm going to be giving a talk at a meeting that considers phage-mediated biocontrol of bacterial pathogens of plants, I might as well do my best to find every single phage-mediated biocontrol of bacterial pathogens of plants paper. And there's actually fewer papers than this looks like, because I got this listed out by pathogen. So some of these papers are up here multiple times, like right here. So there's actually fewer of those. And that's difficult to see, so I anticipated that. And so that goes from 1935 to 2000. I'm sure there's earlier ones than that, but this is what I found so far. And this is 2000 to 2014. Again, some of these papers are in here multiple times. That's it. That's your entire literature, mostly. So never let any kind of task go to waste. If you can turn something into a paper, turn it into a paper. And I fully intend to take the efforts over the past 4 or so weeks putting together this talk and turn it into a paper. In fact, I've already been kind of asked to write a paper for one of these 100th anniversary of Phage's special issues that come out. And I intend to write up what I put together for this talk as part of that special issue. With luck, it'll be accepted. But never turn down an opportunity to write. So I've been blogging So this is the ISBM, the International Society for Viruses of Microorganisms blog. I've been asked by Bob Blaisdell, or however you pronounce your name, Bob. Bob is in the unique position of having been my grad student, having been Betty Cutter, who's very famous in the phage therapy field, not grad student, but undergrad, and now is Rob Lavinia, who's very famous in the phage field as grad student. He's been all over the place, but now he's got this blog, and I contributed to it. And one of the things I found was a paper on how to make phage plaques without using bottom auger from 1963. So it's 50 years old or so. And so anyway, I put together a blog entry for that. So I'm putting together blog entries at the drop of a hat. So I figure that this, maybe not, well yes, as presented here, that will make a good blog entry. I can update these blog entries. I'm not going to update into the future, but if I come across things that are older, if you tell me about older papers, I can go into the past. So going bottomless in the black hole. Never turned down the possibility of having a cute title. So I'm not going to have time to go through these again. That's why I put it at the end. So I could ignore it if I have to. And I'm going to have to. Betty Cutter wants me to make sure that I tell everybody that everybody should be coming to the Evergreen State College for their every 2-year phage conference. And the next one is next summer. And lastly, that was drawn by my daughter. My daughter is attending college, and she has decided that she wants to major in ecology. It's not just me. She thinks like an ecologist. She needs to be an ecologist. But I insisted that she also be an art minor at least. And she was babysitting or bird sitting this conure parrot for my collaborator Paul Hyman this summer in July. And she was bored, and so just started sketching out the parrot, and that's her sketch. Anyway. That's it. Thank you very much.
Okay, thank you, Steve. Very interesting talk.